just give me a shout if it doesn't work. Sorry if there's a little bit of background noise, but it should be fine. All right, yeah, so I'll just jump in. So yeah, so as Danilo mentioned, my group has been working on RNA viruses for a long time, and we started, studying the envelope proteins and entry pathways of envelope viruses, and that's what we were very much doing at Yale when Danilo was in the group. And since then, we've evolved and sort of migrated a bit more towards the host virus interface. And so we're still very interested in focusing on RNA viruses. But our two big questions at the moment that we're addressing are how RNA from enveloped RNA viruses is recognized in the cytosol. And more broadly, how RNA that might be present in the cytosol during a viral infection, it doesn't have to be actually from a virus. It can also be from the cellular RNA. In some cases, you can have stresses or imbalances in the cell that cause certain RNAs to be present that shouldn't be there. And so actually, that's also something we're interested in. But in general, the first question, the question I'll talk about today is how is cytosolic RNA associated with viral infection sensed? And the sensor I'll talk about today is MDA5, and that's really one of the important ones, the key sensors of cytosolic RNA. The second question, just to give you some context that we're working on in the group, is how, what happens when viral RNA may be reverse transcribed into DNA and integrated into the genome. So we're interested in transcriptional regulation of integrated viral DNA, retroviral DNA, lentiviral DNA, but actually lots of different viruses, herpes viruses, integrate their DNA. And we have, of course, a huge fraction of our genome is of viral origin with endogenous retroviruses and retroelements. And we're interested in the cellular machinery that regulates the transcription and gene expression of those sequences, of those integrated viral genomes, and also in the really interesting and important examples of when those genes escape repression and acquire useful, sometimes essential functions. Functions can be essential for life that are fulfilled by proteins of viral origin. So that's kind of where our direction has moved towards. But today I'm going to focus pretty close to the virus itself, to infectious viruses, and really talk about how viral RNA and viral associated RNAs are recognized by MDA5. So MDA5 is a cytosolic sensor, as I said. It's one of the key sensors. And it really, what it senses is long double-stranded RNA, and that's a viral signature of infection. So a signature of viral infection. So it can be from just genomic RNA just delivered, but actually you can also get double-stranded RNA as replication intermediates. So it's not necessarily just freshly delivered, but it's made in the cell by the virus. And actually, you can also get cellular RNAs such as allele repeats or incompletely spliced transcripts that make it to the cytosol. And these are danger signals that are maybe of cellular origin, but that are a sign of infection and that are still sensed by MDA5. So MDA5 is actually broader than just a viral RNA sensor. It's a infection-associated RNA sensor. So we're really interested in how it works. And our group and other groups have studied MDA5 for a while, and we know that it assembles into filaments on RNA, cooperatively forms these filaments. And in doing so, it creates a platform through its caspase recruitment domains, card domains, to recruit the downstream signaling molecule MAVs, which then forms its own fibrils. And MAVs is anchored on the mitochondrial membrane. And this signaling complex, as you can see, it's quite on the right-hand side of the slide. It's quite large and complex, and it serves as a platform for recruitment and amplification of a strong interferon beta response. So you get a strong inflammatory response in consequence to this RNA sensing. So. A notable thing about MDA5 is that when things go wrong, that you can have clinical outcomes. So it's a hot spot for clinically relevant disease mutations. And there are actually many genetic variants linked to different clinical outcomes, diseases in MDA5. And so this is from the latest ClinVar database down here on the bottom left, 1,685 clinically relevant annotated variants, genetic variants identified in this one gene. And that's actually a very high density. If you can take your favorite gene, most genes have a lot less, many fewer variants per nucleotide than MDA5. So it's a real hotspot. And that's because sensing RNA is dangerous business, right? That there's a lot of RNA in the cytosol that should be there, transfer RNA, ribosomal RNA, mRNA. And if MDA5 or other immune sensors recognize those by mistake, then that you're going to get an autoinflammatory response, which is toxic. And so when things go wrong with mutations, that's when you get autoinflammation. So I'll talk quite a lot about our work on different clinical variants of MDA5 and the different molecular mechanisms of disease associated with those. But first, just to take you back to the, how our first studies on MDA5 were really to understand how it forms these filaments, and because that's the on switch, the activation mechanism we knew from previous work by our group and other groups for MDA5 is this filament formation. And so, because the filament formation depends on a helical filament interface, we wanted to understand what that looked like. And so we applied cryo-EM imaging, and this is Chinyu, who collected thousands of these electron micrographs where you can see 8 to 10 nanometer filaments of MDA5 bound to RNA. You can see a few regions of naked double-stranded RNA as well. And these filaments are quite well ordered, and you can average many thousands of filament segments. And through the power of image processing, cryo-EM image processing, we're able to reconstitute determine the structure of MDA5 bound to RNA at sufficiently high resolution that we're able to build an atomic model of the complex. And that's what we're looking at here. At the center is the double-stranded RNA, and here we have 3 subunits of MDA5, and the interface between each subunit is the filament-forming interface that's absolutely essential for MDA5 signaling. So mutations at the interface inhibit signaling. And so this structure really showed that very nicely. But what we were also able to do is soak different nucleotides into MDA5, because what I didn't mention is MDA5 is actually an ATPase and hydrolyzes ATP. So we were able to take ATP, ADP, AMP-PMP, a transition state analog, and soak these into our sample and then determine structures individually. And if I just morph between the different structures with the different nucleotide bound states, we've got AMP, PMP, and ATP and ADP here. You can see there's actually quite a lot of rearrangement and changes in how the RNA is positioned relative to the protein in the different nucleotide states, right? And so this got us thinking, what's actually going on here, and what is the ATP hydrolysis cycle being used for in this complex? And the hypothesis we came up with is that it's actually ATP hydrolysis. It's clearly driving changes in the structure of the protein and in how it engages with the RNA. And the idea is that it's actually challenging the interaction, and so that if it's really a double-stranded RNA, it can stay on. But if it isn't a perfect double-stranded RNA, the ATP hydrolysis cycle should make it fall off, and it will act as a mechanical proofreading mechanism. So this is like this idea is shown schematically here, and this is actually based on the structures that we determined with the different nucleotide states. We can see that they have different helical twists, and also they have different RNA footprints. And so when ATP binds to the MDA5, the protein engages pretty tightly and binds pretty tightly on the RNA with a 14 base pair footprint. And then when ATP starts becoming hydrolyzed, this is the transition state analog, ADP aluminum fluoride complex, the footprint increases to 15 base pairs. So the protein's slightly expanding on the RNA, and it's also gripping even tighter. We measured that based on the protein RNA interface, the buried surface area between the two species. And then when the hydrolyzed ADP dissociates, the grip on of the protein on the RNA relaxes a little bit, so that it becomes a little bit weaker binding, and then it relaxes even more in the nucleotide-free state, in a different type of nucleotide-free state. We call it the high twist state because it has a higher helical twist. So what's going on here is that you have a cycle of ATP hydrolysis coupled with conformational changes, and the tightness of the complex is changing, and it's loosening at one stage of the cycle. So we think this is giving an opportunity for dissociation of the RNA, of the protein from the RNA, during the ATPase cycle, and that if the protein is a viral RNA, the double-stranded RNA, or the abnormal RNA, it will stay associated, and if it's a mismatched RNA or the type of RNA that's commonly found in a healthy cell, it will be more likely to dissociate. So it's a mechanical proofreading mechanism. That's our hypothesis. And so this would make a prediction, is that if you lose ATPase activity, then in the enzyme, in the protein, right, in MDA5, then you should lose the proofreading and you should start getting signaling in response to non-ideal or imperfect or cellular RNA ligands. And that's actually what happens. I'll show you that. But first, let me just introduce our disease variants. I just mentioned those earlier. There's various different types of disease-associated variants that have been identified, and the first ones to be identified were all auto-inflammatory. So they cause different kind of chronic elevated interferon conditions or interferonopathies. And these mutations map to either to the RNA protein interface, or they map to the ATP binding site. And with one exception here, this one I'll talk about more later in my slides, which maps to close to the filament interface. And actually, what all of these mutations do, we think, is that they're interfering with the protein binding to the RNA directly, by weakening or by strengthening or weakening the RNA binding interface, or they're changing the ability of the protein to hydrolyze ATP. And I'll show you some data that confirms that in a little bit. And then, so that's one category of mutations. But then there's another category which is not linked to autoinflammation or elevated interferon. But they were shown in, these mutations were shown in different GWAS studies to be protective in type 1 diabetes. And so this is a bit strange. This is an autoimmune disease, of course, and the mechanisms were unknown. And so this is something we wanted to look in more detail. There's these 4 variants here, all associated with lower risk of type 1 diabetes. And so we think that, that's a completely different category of, or at least clinically, it's definitely a completely different category of mutations. So next we looked at, we've teamed up with a scientist here, a bioinformatician, Chris Wallace, to look at GWAS. These are genome-wide association studies in different autoimmune diseases and also in inflammatory bowel diseases, to ask how these mutations and how these T1D protective mutations affect T1D, but also other diseases, because there were some publications suggesting that the protection was not limited to type 1 diabetes and these MDA5 mutations, but that it extended to other diseases. And this this is exactly what we found. So here's 6 different variants of MDA5. And these effects here, these graphs are showing the increased or decreased risk of patients having the disease. And so type 1 diabetes, it's always negative odds ratio, which means that there's protection. And same for psoriasis and hypothyroidism. And all 3 of these are autoimmune disease. And this is true of all 6 variants, but to different extents. So this is the most protective here, the top left, and bottom right is the smallest magnitude, but in the same direction. What's interesting is that the inflammatory bowel diseases, Crohn's and ulcerative colitis, actually there's increased risk. The odds ratio is positive. So if you decrease the risk of autoimmune disease, it seems like you always get increased risk of IBD or gut inflammation. And so it seems like this is kind of a signature of a trade-off. You win something, but you lose something, And because it was such a strict correlation, always protection against autoimmune and increased risk against gut inflammation, we thought that they were probably linked. I'll just show you another graph related to this one in the next slide. And this is showing you that the 6 different mutations I showed, the degree of protection or increased risk is correlated with the frequency in the population of the mutation. So this one, splice 1, this variant, which we called splice 1, is the most protective against autoimmune diseases. It's one of the ones that increases the risk the most of the IBD diseases, but it's also the rarest in the population. It's present in like 1% of the population. These on this other side, T946A and R843H, they're extremely common in the population. In fact, it's almost like 50-50, and there are geographic variations. So, in Africa and Europe, the dominant variant might be different. So R843 might be the dominant variant in Asia and Africa, whereas H843 might be the dominant variant in Europe, So, and accordingly, the degree of protection are, an increased risk against the diseases is much smaller. So these are mild mutations. They have a small effect, but they're very common in the population. And the ones that have a very large effect are rare. So you could kind of almost guess that, the rare ones are probably kind of dangerous or not so good to have, and that's why they're rare. And the common ones, there may actually be an advantage, and that might be why we see different geographic variations of the common ones in some places. For example, if you have, a Western diet, you might be more susceptible to gut inflammation, and these ones might be more harmful if you have increased risk of gut inflammation, and you might have a lower viral disease burden, and so it might be okay to take a trade-off. So that's what we think is going on. That's just a hypothesis. But just to spell out the hypothesis a little bit more explicitly, we think that these are actually loss of function MDA5 mutants. And in fact, one of these mutants is a truncation of a large truncation, so that kind of makes sense. And that these loss of function mutants, the loss of function is to different degrees, but they always protect from autoimmune tissue damage, for example, to pancreatic beta cells, islet cells, and that might lead to type 1 diabetes, right? And so that's why these are protective against type 1 diabetes, because you're less likely to have MDA5 dependent inflammation in the pancreas. But the flip side is that you have an increased risk of persistent infection, for example, in the gut, and which could cause increasing incidence of IBD. So that's a hypothesis, but it's in agreement with the GWAS studies. So we wanted to understand a little bit more mechanistically, what's happening at the molecular level, and why are these mutations having the effects that they have. So we started picking through selected mutations. One selected one here from the autoinflammatory variants, and then I'll show you a couple from the type 1 diabetes protective variants. And so this one, M854K, that's the interesting one that didn't fit in the ATPase active site or in the RNA protein binding interface. It actually maps to the filament formation interface, M854K and MDA5. And if we look at this signaling assay, this is an interferon beta reporter assay where we're measuring, interferon beta promoter activity in HEC293 cells, so a very standard signaling assay. We stimulate the cells with poly-IC double-stranded RNA or no RNA. And with wild-type MDA5, you only see signaling with the poly-IC, as you would expect. But with M854K, you actually see signaling even without adding double-stranded RNA to the cells. And so what this means is that there's signaling occurring in response to cellular RNA, right? Because we haven't added any exogenous RNA and there's no infection in these cells. So the signaling must be coming from endogenous RNA. And this is telling us that M854K it's failing in the proofreading, right? It's not, it's signaling from RNAs that it shouldn't signal from. So that's at least what we found there. And this is very similar to what you get in ADAR knockouts. ADAR1 is a really interesting and important enzyme that goes around the cytosol modifying RNA. It modifies adenosines to inosines to make them actually less visible to MDA5, cellular RNAs. If you knock it out, you have chronic inflammation, and it's actually lethal in mice, but it's rescued by MDA5 knockout. And so what's going on is that ADAR is masking covalently changing all cellular RNAs so that they don't activate MDA5. When you knock out ADAR, you get constitutive MDA5 dependent signaling with wild type MDA5. It's with this M854K, it's still the same, but that happens even without the ADAR knockout. So that's what constitutive baseline signaling looks like, and I think that's why gain of function mutations cause elevated interferon. It's because you have signaling from endogenous RNA. Now, just to take a look at the signaling activities of some of these autoimmune protective, T1D protective variants, I'm just going to show two of these for the moment. This is that truncation variant, E627, and then a premature stop codon, and I923V. Both of these reduce the signaling. This is actually a different assay, but it's the same kind of idea. Both of these reduce the signaling to baseline or even below baseline in this case, relative to wild type. And wild type has tenfold higher signaling activity. This is actually cells that are infected with virus because the poly-IC is slightly artificial, so we wanted to also make sure that this worked with in a more physiological screen. So this is a RT-qPCR assay with EMCV virus. So these two T1D protective mutations, we conclude are loss of function in terms of signaling, right? So we've got autoinflammatory variants, gain of signaling function. Autoimmune protective variants are loss of signaling function so far. But remember, it's an ATPase, the MDA5 enzyme, and so we also measured ATPase activity, and here we got a surprise. Or at least one surprise. Some things were expected, some things were unexpected. It was as expected, the truncation mutant 627, which is a T1D protective, autoimmune protective, is completely dead. No ATPase activity. Okay, loss of function. But the A946T actually had wild type ATPase activity, and I923V, which is also T1D protective, actually had 3 times higher ATPase activity. So it's hyperactive. So we're seeing different phenotypes here. And in terms of the other types of mutations, the autoinflammatory ones, the ones that cause elevated interferon, so these are the gain of signaling function mutations, these are completely ATPase dead, at least these two important ones that have been, the one I just mentioned and another one that was looked at in the literature previously. And so actually ATPase activity, you can have various effects on different mutations can have different effects on ATPase activity. But just remember that the I923V is hyperactive, because I'll come back to that, because we can actually now understand why that is. And the first big clue is in filament formation. So remember, the activation switch for MDA5 is forming filaments, right? And so we looked at the capacity of the different clinical mutations to form filaments on RNA. And with M854K, we looked with relatively short, double-stranded RNA, 100 base pairs, because wild-type MDA5 does not recognize RNAs that short. It really senses longer RNAs. It's specific, has a length threshold. And so that's part of what it does. So that's so it doesn't recognize, random cellular 100 base pair allele repeats, for example. So you don't see filaments with wild type, but with M854K, you do see filaments. You see short filaments that are the same length as a 100 base pair RNA. And so this is really demonstrating that the M854K, and remember, M854K's ATP is dead. And so this, I think, is some experimental evidence that the protein is failing to proofread. It's binding to RNAs it shouldn't bind because they're too short. Wild type does not bind these, but it is binding to the filament, to the RNA, and forming filaments. And we think that's because its ATPase is dead. It's lost ATPase activity, and you don't have that ATPase hydrolysis cycle to challenge the interaction and cause MDA5 to fall off the filaments. And so that's why you get signaling with cellular RNA only with this gain of function variant. And then conversely, with the loss of function T1D protective variants, you see, as you might expect, a loss of filament forming capacity. In the case of 627 stop, it's a total loss. You see no filaments at all. So this is complete loss of function. And so it has no ATPase activity, but that doesn't really matter. It doesn't even form filaments in the first place. And so that's why, it's a loss of function, signaling function mutation. I923V also has less efficient filament formation. It's a more subtle effect, and we quantified it in this paper, but it is a statistically significant effect. The I923V forms shorter filaments on average. So it's a milder mutation, but it's definitely there. And remember, this is the one that's hyperactive. This one has more ATPase activity. And so what we think is that it's actually an overzealous or a sort of overactive proofreader. It's falling off RNAs that it shouldn't fall off. Actually, I forgot to mention this. These are on the right-hand side. It's 1 kilobase RNAs. So that's why wild type does form filaments because these are virus-like RNAs. They're long ones. And so you have a partial loss of filament forming capacity with I923V that we think is related to the increased ATPase activity, which is causing it to fall off prematurely. And then actually A946T, we really couldn't see much difference or any difference to the wild type. But that's actually the one that's the mutant that on the graph with the effect size had very small effect size, and it's the very common mutation. So it's a very subtle effect, if any, and we couldn't see it in this assay. So next we wanted to look in even more detail, what's happening at the molecular level. And so we applied our cryo-EM reconstruction method, to ask now, what happens in these different mutants and what effects are the mutations having on the structures. And so this is 854K, the gain of function mutation, the interferonopathy causing mutation. And this is the lysine that is present in the mutant, right? And this lysine is actually forming really tight interactions, polar interactions, a salt bridge and a hydrogen bond with other side chains in the protein. And what that's doing is it's kind of locking it in, and it's constraining it, so it's causing a restraint that that we think prevents ATP hydrolysis, right? Because remember, ATP hydrolysis, you get these conformational changes occurring that accompany the hydrolysis. So they're actually necessary for hydrolysis. So when you impose these polar bonds here, these polar tethers, you can no longer undergo the conformational changes, and you can no longer hydrolyze ATP. And so that's why this is a ATPase dead mutation. Even though this mutation is very far from the ATP binding site, it's an allosteric mechanism for inhibiting ATP hydrolysis. And now this is the wild type, just for comparison, right? Wild type has a methionine, and it's much looser. There's no constraining polar interactions, right? So that's how I think we understand in detail. And then just to test this, we created additional mutations. These polar contacts are with serine 491 and glutamate 813 and this is the natural variant M854K. So if we mutate one or the other of these partner residues, actually that you still have no ATPase activity. But if you mutate both, so if you mutate S491 and E813, you completely recover ATPase activity, even with the disease variant there. So you've got M854K is still present, and that really demonstrates, that proves that it's really these constraints, it's really these polar bonds that are preventing ATP hydrolysis. Because when you lose the partners, you recover activity. So yeah, I think that really nicely showed, I think, how this disease mutation is working, and it you get the same result actually if you have a mutation that inhibits ATP binding, but here we have a mutation that inhibits the hydrolysis in an allosteric mechanism, and in both cases, it's a loss of signaling function. A gain of signaling function and a loss of ATPase activity. Due to the stabilized filaments, due to the lack of ATP hydrolysis. Yeah, and this slide was just showing, just proving the point, with M854K, it fails to expand the footprint from 14 to 15 base pairs. You have a mixed population of 14 and 15 base pair footprints with the aluminum fluoride ATP analog. So this is just showing that conformational change that should happen with ATP hydrolysis is not happening with ATP hydrolysis. So that's just additional evidence. And also we were able to show, that you can plot the helical twists, and you can see that there's perturbations and changes in the helical twists with M854K, which again demonstrates that the changes in twists that normally accompany ATP hydrolysis are defective in this mutant. So it's really an allosteric mutant. Now, just to switch gears a little bit back to the T1D protective mutants, this is our more recent work. I923V, remember that's the ATPase hyperactive mutant that's T1D protective. And so what does that look like on the structural level? We solved several different structures with different nucleotide analogs. And actually the protein is pretty much fine. It's a very subtle mutation from isoleucine to valine. There aren't any big changes but so we really had to drill down on the details. So at this level, they look kind of pretty normal. But then when we looked in more detail and really zoomed in to the site of the mutation, this is the wild type residue I923. This is the mutant residue V923. And what you see is actually a very subtle, a very slight change in the interaction with the RNA. So this glutamate, instead of really contacting the RNA and forming a contact, you lose that contact with the ribose group in the RNA. So it's just slightly weakening the affinity to the RNA. This is a very finely tuned protein, and because it binds to RNA very cooperatively, a slight decrease in affinity is actually enough to significantly destabilize the filaments, and that's what we see here, and that's why N23V, we think that's why it formed shorter filaments, and so that's why it's a loss of signaling function, because the filaments are shorter and less stable. This other mutant 946 also looks pretty much just like wild type, and it looked like wild type in the filament forming in the ATPase. So we just conclude that might be a post-translational modification, or the effect might be so small that we just don't observe it in any of our in vitro assays. Okay, so that's just to summarize. We've got, for the T1D protective mutations, we've got different mechanisms or effects on the protein. We have hyperactive ATP hydrolysis. That was I923V. We've got 627 stop, which just basically causes the protein to destabilize. It doesn't form filaments at all. It's just completely defective. So that's a really sort of traditional loss of function mutation. And then this 946, which has a very subtle mutation, or a very subtle effect, which still remains to be determined. Our R843 actually, in the meantime, we've discovered that it's non-causal because it's genetically linked to 946, and that's why it's being picked up in the GWAS. So just to summarize, you can have various different clinical outcomes depending on what the effect of the mutation is in MDA5. And this is why it's a hot spot for disease mutations, this gene. Mutations that cause loss of RNA binding, decrease of RNA binding affinity, or gain of ATPase activity. Both of those things result in less stable filaments and decreased interferon output, and therefore increased viral load and possibly increased risk of IBD. But you may also be protected against autoimmune diseases that are a result of MDA5 signaling. And conversely, if you gain gain RNA binding affinity or lose ATPase activity, you get more stable filaments. And then this is what causes increased interferon signaling, because you need the filaments for interferon signaling, decreased viral load, but you have autoinflammation. So, you might be glad to have lower infection rates, but you have more inflammation, and this can cause problems, and if it's chronic. So it's all about a trade-off, and the protein is really carefully tuned for the environment. And that's why we think different environments might mean that different disease mutations are actually more favorable than others if the effect is mild. Okay. I see I don't have much time, but I just have a couple of slides left, and I'll just briefly mention, this little vignette here, and then I'll wrap it up with time for questions. So I just want to mention, one question we asked about ATP hydrolysis with MDA5 is whether it's cooperative. So if one subunit hydrolyzes ATP, does it make it more likely that the neighboring subunits might hydrolyze ATP, relative to a subunit far away in the filament? And that's because of the conformational changes. You might imagine that they might spread to the next subunit, that you'd get a chain reaction of ATP hydrolysis. And Rahul Singh in the lab checked this in a clever experiment where he mixed wild type and mutant ATPase dead MDA5. And this ATPase dead mutant forms more stable filaments, but doesn't hydrolyze ATP. And what we saw is that when you titrate in more and more of the ATPase dead MDA5 into the wild type mix, you get a steep decrease in ATPase activity of the filament. And if you didn't have any cooperativity, you would expect a linear increase or decrease. And, there's various controls for that. But that's a well-established mix. Linear titration of ATPase dead mutant should give you linear decrease. So the fact that the curve follows this more exponential shape is really showing that actually the ATP hydrolysis within the filament is cooperative. And the reason that's interesting is that because, if you have cooperative, let me just step back. If you have cooperative hydrolysis, remember each hydrolysis event is coupled with a increase in the RNA binding footprint. And so if you have a chain reaction of these, you're going to keep increasing the footprint. And because you have a continuous filament, the footprint of the filament eventually is going to increase by quite a bit. And so there's a possibility that you might be able to displace RNA bound proteins that might be present on the filament. And so then we did a protein displacement assay. This is still Rahul in the lab did this, where we were measuring displacement of streptavidin from an RNA as a function of MDA5 filament formation and ATP hydrolysis. And just to cut a long story short, MDA5 can displace streptavidin from biotin on an RNA. It's a very tight interaction. But if you form continuous filaments all the way up to the streptavidin and then add ATP, you get displacement of the streptavidin. And so this is free RNA. And so here you have mostly free RNA with wild type MDA5. If you don't add MDA5, you have very little free RNA. But if you kill the ATPase activity with this mutation R338G, then you lose, again, you lose the displacement activity. If you kill the filament forming capacity with this triple mutant, you lose protein displacement activity. Generalizable ATP analog with wild type MDA5 also, you don't have displacement activity. So we have an ATP dependent displacement activity. And we think this is could be useful for MDA5 to displace, viral proteins, for example, bound on viral RNA. And it gives it an additional antiviral activity. And that's shown here. So you got sequential hydrolysis events leading to displacement of a viral protein or a bound protein from the RNA. Okay, yeah, and just finally, we have a collaboration with David Dulin looking at some biophysical approaches, very clever and elegant approaches developed by David's lab, magnetic tweezers. But I'll just refer you to this paper if you're interested, because I think we're out of time. And so with that, I'll just thank everyone in the lab who participated in this project, the Rahul Singh and Joe Joyner, and former lab members Alba and Chin played instrumental roles in the MDA5 project. And now we've got Madeline Eaton has just joined and taken the relay. Chris Wallace did the bioinformatics, and Frank van Kuppenveld did the EMCV assay. I mentioned David. And thanks to funders and thanks to you for your attention. And then let me show one last slide, which is, this is the MODIS group in 2025. This is the MODIS group in 2018. And here we have our friendly host, who's looking pretty happy to be in the New Haven sunshine. So yeah, thank you very much. And I'll take any questions.
That's great. So great talk, and also great research. So it's an amazing, yeah, that photo is something. So the microphone is on. So if anyone has any questions, so you can ask to Professor.
Okay. I'll stop presenting for now, but yeah.
Yeah. So I have one question. I have several questions actually. But one question is, have you performed any viral infectivity assays in those cells that have those variants with the MDA5, just to see if you have like increased viral replication or antiviral responses against infection?
Yeah, so the assay with the MCV, that was an RT-qPCR assay with Frank van Kuppenveld, and that had MDA5 knockout cells where we were adding back in the mutants. So it's not exactly it's not the endogenous locus, but we were looking at, and we weren't looking at viral load. We were looking at transcripts. We did look at viral load actually of EMC, and that seemed to be less variable. But I think that depends a lot on the time point chosen, and so. Yeah, we didn't see an effect with EMCV in that with the MDA5 knockout cells complemented with the mutants, but it would be nice to do it with CRISPR or actual maybe even patient cells or primary cells or CRISPRed cells with the endogenous locus, and to really look at different and perhaps more other types of viruses as well to look at viral load. We haven't done that.
Okay, great, Professor Rodrigo.
Thanks, Yorgo, for the talk. It was wonderful. I wonder, you brushed shoulders with the influenza virus a long time ago. I don't know if you still study flu. Present, do you? Do
we study flu now? No, we're not studying flu at the moment. But yeah, I think it's an important one, right? Because that's one of the likely things we have to worry about in the near future. I think flu is maybe close to the top of the list. So
I was wondering if what do you think of these variants of MDA5 participating in the persistence of influenza virus? We have seen persisting flu in lymphoid tissues from patients without symptoms who undergo surgery for tonsillectomy, and we find inside different types of cells, including CD8 cells. But since the flu virus replicates in the nucleus, And I don't know, what do you think of MDA5 as a participant in this?
So yeah, I think it's possible. There could be an interesting link, and I think just in general, checking the MDA5 genotype of a patient could be informative with in context of viral infections and viral status. With flu, it's not the most. MDA5 is not the main sensor for influenza because, as you say, the replication is mostly shielded from the receptor and the cytosol, and there's the capping, and so I think maybe Regai has a slightly larger role, or and then there's other RNA sensors, PKR and others. That might also feed in. So flu is not one of the viruses that are most strongly sensed by MDA5. It just managed to mostly escape or hide itself. But there could still be an effect. I don't know. Yeah,
how easy is it to do the genotyping for MDA5?
Well, I can't really tell you because we've just been tapping into these large data sets that the GWAS data sets and the biobank. The UK is we're fortunate that we have a lot of genome sequences and access to patient sequences, and they easily do whole genome sequencing. Especially if there's a chronic condition. So, but yeah, I think it should be relatively straightforward. It's not in a complicated, position in the genome or anything. But I don't have personal experience doing the genotyping with MDA5.
Okay.
So
it would be interesting to see if the genotype correlates with the persistence. Okay, thank you.
I think so. Yeah, we've been. I don't know if flu is might be a tougher one to start with, but I think in general that's a really good idea to check correlations like that. Yeah.
Thank you.
Okay, Orlando.
Hi, great talk. So I was curious about your mutant TM854K, which increases interferon beta expression even without poly-IC. So my first question is whether you showed that you have this activation and formation of sharp filaments even without other stimulus. And this is kind of different from your control with ADAR1 knockouts, because when you don't have ADAR1, you have more formation of endogenous double-stranded RNA. So I was curious whether you try to rescue activation of interferon beta by this mutant by overexpressing other dsRNAs, because there's some literature showing that when you overexpress ADAR, PACT, etc., and many other dsRNAs, you can actually kind of buffer the activation. So did you try it?
That's a good question. We haven't tried that. ADAR1, it's and MDA5, but ADAR1 in particular has attracted a lot of interest as a target, as a drug target. But it's very difficult to work with, biochemically, it's very difficult to work with, to isolate, especially the P150 isoform, which is the one that's relevant in inflammation and human disease. So we haven't been able to do anything, but yeah, I think it's a good suggestion to look in cells, and maybe it can be overexpressed in cells and see what effect that has. But no, we only just tried with a knockout. But I think you should see these things are tightly linked, and that's why the MDA5 knockout is because it's a complete rescue of the ADAR knockout. You return back to baseline. So I think it'll be dose dependent very much, I would expect. And another complicating issue here is that in patients you might have two alleles and with different different alleles, different variants on each one, and so that can make things complicated as well. So we're oversimplifying things in that sense as well in our experiments. We're not looking at mixtures of different variants, but that's a good point. Yeah, we should try titrating ADAR1. It has to be done with the P150, I think, though. P150 isoform, probably. Yeah.
That's true. A second question is whether you try to see twitch RNAs isoforms the mutant is binding in the cytoplasm for the formation of the short filaments by rip or clip assays. And if you also try to see what is the minimum size of the filament, because as you said in the beginning of the talk, MDA5 binds more long filaments, and this changes between the dsRBP that you're looking at. PKR is more short filaments, right? So you just try to see what is the minimum filament size and the minimum size required for the activation.
That we didn't show that, but we and also others have looked at the length dependency in some detail. So so there's a pretty sharp threshold. It does depend on the ATPase activity, so it depends a bit on the variant, right? I showed that some other variants bind to shorter segments, but wild type, you know, at the baseline, you need more than 100 base pairs, and so at 150 or so, you start getting binding and signaling and filaments stable, and then but it continues getting better until a kilobase, and it's really a kilobase is the maximum. But anything from 150 to a kilobase, you're on the curve, on the increasing curve. If you remove the ATPase activity, you can reduce the length threshold, so you start getting binding of smaller things. You know, so it's dependent, and it's also dependent on how many filaments you have. So if the protein is very dilute, it's binding just as monomers and dissociating, and so then, you know, there's no length dependence to that. So you really need to be in the filament forming regime. So it's a relatively complex thing, but yeah, we've mapped it down to, you know, a pretty sharp increase from 150. But the other thing is, you know, there's a lot of modifications on RNA. So ADAR adds inosines, but of course there's a whole epitranscriptome, right? And so there's 150 different chemical modifications, I think, that can be added to RNA. And actually not all, but many of them affect in one way or the other way the recognition. And that's something we've really only started to look at. I mean, there's a little bit of data out there, but that's a big area of research. You know, as you know, like the COVID vaccine was, you know, it's using pseudouridine, and like one reason is that it helps escape things like MDA5 signaling and also other RNA sensors. So, but that's not a common modification in nature. But uridine is, of course. So, yeah. Oh, sorry, pseudouridine is common, but the methyl pseudouridine that they use is synthetic, I think. Anyway, so yeah, so we would like to look at covalent modifications in more detail for sure.
Thanks.
Okay, thank you, Orlando. Do we have time for one more question?
Yeah, one more question.
Yeah, there's one here in the chat box. Jonathan is asking why displacing double-stranded RNA-bound proteins is important in the context of MDA5 function, if proof reading is made by the length of the RNA molecule. Also, can you comment on what makes double-stranded RNA tolerable, human versus virus?
I didn't quite get the second part, but the first question, the first part is relatively straightforward. So a lot of viruses encode RNA binding proteins. They might be something that binds to the cap and shields it and protects it, or something to attract the polymerase, or something just to package the RNA and protect it from proteases, or protect it from RNA sensors. So there's a tug of war going on there, and you know, if the cellular protein RNA sensors can come in and displace those viral proteins, then you know that's bad for the virus and good for the cell. There's an example, I think it was VP35 from Marburg, or the filoviruses, which is you know, binds to RNA and can be displaced, I think with difficulty by MDA5. So so that's just a direct antiviral activity, displacing the viral encoded proteins. The second was about the cell versus viral RNA. Could you just repeat the second part, Danilo?
Can you comment on what makes double-stranded RNA tolerable, human versus virus?
Yeah. So in humans, actually humans don't have long stretches of double-stranded RNA, and so you know, one of the biggest sources of double-stranded RNA in the cytosol are these allele repeats. these retroelements that are found in non-coding regions. They can be found in some mRNAs and the 3' end or in introns. But even though they can be relatively long, they're rarely longer than 150 base pairs, but they always have some mismatches and loops, so it's not a continuous duplex all the way through. And the longest continuous double-stranded stretch is 30 or 40 base pairs in an allorepeat. And so there's not that many there's not that, ribosomes are tightly folded, tRNAs are tightly folded. So there's actually just not that many sources in the cell of, it's kind of surprising, but it's not a common signature to have continuous stretches, double-stranded.
Okay, that's great.
Great.
Thank you so much for your time and your talk. And
thank you.
That's amazing, and it's great to see you again. And you can
great
join
to see
us
you too
anytime you want. So we are always
yeah. Let's
here.
catch up at a meeting, or I hope to see you soon. Yeah.
I need to visit you, and you also need to visit us.
Yeah,
here
let
in
me
Brazil.
know if you're in London. Yeah.
Okay, that's
Great.
perfect. Thank you. Bye.
Thank you, everyone.